Okay, next we have Nick Sard, who will be talking about environmental DNA metabarcoding sampling methods.
Hey, thanks everybody for sticking around for the last talk of the meeting, at least in this room. Today I'll be talking to you about environmental DNA metabarcoding and how we assess its effectiveness for detecting low abundant species in aquatic communities in Michigan. I am actually a new hire at SUNY Oswego, and I wanted to give this talk really just to demonstrate one of my tools in my tool set that I'm hoping to apply in the Great Lakes Basin. But this work was funded by the Great Lakes Restoration Initiative through the Michigan DNR, and I'm coming from a postdoc at Michigan State University working in Ken Scribner's lab. So this is a classic invasion curve where you have two y-axes. The first is area infested, the second is control cost, and the x is time. And the point of the figure is to say that eradication and containment are much more feasible in smaller areas. And so naturally, early detection is a top priority for management agencies. And in Michigan, they use their standardized statewide status and trend survey to survey for fish communities, but also as a tool for aquatic invasive species detecting. And the question is, how effective is this method? Now, traditional gears have been evaluated for maximizing early detection of aquatic invasive species in the Great Lakes harbors, and that's been implemented by the U.S. Fish and Wildlife Service, but little work has been done on inland lakes. And we were interested in layering in an eDNA metabarcoding component to this survey for aquatic invasive species. So most of you probably know that plants and animals constantly extrude DNA from themselves into the environment, and that we can use standardized regions of DNA to act as barcodes. These standardized regions of DNA have regions of conservation on either end of the locus of interest, and there's a lot of variability inside of that locus. And that variability can act as a barcode for various species. That is our 12S DNA database. And we also have one for 16S for fish. So the goal here was to evaluate the effectiveness of the status and trend survey to monitor for aquatic invasive species and assess if there's an added value to adding a eDNA component to this. So Michigan randomly samples lakes of different size classes throughout the state each year. We chose 8 of those lakes, and within those lakes, these state fisheries division will develop a stratified random sampling regime using multiple different gear types with the goal of sampling the entire fish community. So after they did that, we'd go into the same lake and collect about 50 water samples, and in total over the 8 lakes it was about 400. I have some Google screenshots of the different lakes that we sampled, and the point is that there is a lot that we don't understand about the ecology of eDNA. And that all of these systems are different. Some are urban, some are rural, some are directly connected to the Great Lakes, some are isolated, some are large, some are small, and it's important to assess the ability of an eDNA approach to characterize the fish community and the rare species within that across all these scales. So just as an example, one of the lakes that the DNR sampled was Dumont Lake. And what I have here is just a picture of the lake, and each dot represents a location that the traditional guys went in and sampled, and the colors there represent the different types of gear. And then we'd go in about a week later and sample that same lake. And sometimes we take water samples, and sometimes we use a Van Doren sampler to collect water from the vent. And that's just a overlay of the two different approaches with the idea that this is what we perceive as a relatively comprehensive sampling regime for that system. And this is just my little pipeline for eDNA processing. So you take a water sample, extract the eDNA, you use your locus of interest, amplify that across many different species, and process that information, or sequence all of the amplicons that you produce using high throughput sequencing technology, process the sequencing data. Usually off of an Illumina run you have like 10 million sequences using just standard bioinformatic process or pipelines, and at the end you can identify specific sequences back to their species. So this is a community matrix that I created for one of the lakes we sampled, and they're all very similar. So columns are species, rows are sampling efforts, and the color there represents the relative number of sequences allocated to a given species, with the gray cells being species we didn't observe in a specific sample. And this is a community matrix based on traditional gear sampling. And just a couple things to point out. One, common species are seen across both approaches. But there are several cases where a species was only observed in one or two samples for traditional gears, but eDNA approaches sample that species in the vast majority of cases. Now, with the eDNA metabarcoding approach, a conversation in the field that we're having is to what degree of uncertainty and stringency do we use? Like, what is real? And I kind of had that comment to Scott, who was talking about gobies earlier today, about this topic. And I think what we have ended up landing on is that if we observed a species in at least two samples, or with at least two different markers, then we considered it to be a real observation. So the figure, well, the text is a little small, but this is another way to look at our data, where each row here represents a species observed, and each panel is a lake, and the columns there represent the 12S locus, the 16S locus, and the traditional gear. And what I am showing here is that the vast majority of times where traditional gears observed the species, we observed it in both loci. And there are several cases where it's observed in at least one locus. And then also there are some species that are observed consistently using either a single locus or in multiple samples, or two loci in at least one sample each, that were only observed using the eDNA approach. One thing that we did find in our data is significant correlations between species occupancy as measured using eDNA, so that's the proportion of samples where a species was observed, and that species rank, so that's based on abundance using traditional gears. And that's across both loci and in the vast majority of lakes. But there are cases where that's not true, like Fourth Lake or Heathcote Lake. And that on average, the mean number of species observed per the 12S, 16S, and traditional gear approach for each lake, most of the time the eDNA approach can sample more species in a single sampling effort. And you can see that in species accumulation curves with species richness on the Y and number of samples on the X for the different lakes. So that's true sometimes, that the slope is steeper for the two different eDNA approaches and asymptotes higher in those approaches compared to traditional gears. But again, it's not always, it's relatively consistent, but not perfect. That is, in Holloway Reservoir, there's a lot of overlap, or the same is true for Fourth Lake or Walloon. Now, one question to ask is, do eDNA methods provide a more complete picture of the fish community that we're looking at? And so you can estimate the total number of species in a community using chow or jackknife species estimators. And we did that using traditional gears alone. And then we looked to see if the total number of species observed using environmental DNA were within those 95% confidence intervals. And in most of the cases, the answer is yes. And in some cases, like here in Heathcote Lake, we're observing more species than would be expected just based on the traditional gear. So I kind of think of that as maybe we're sampling something more than just the lake, something within the watershed. And another approach in terms of thinking about, well, how many eDNA samples would I need to characterize a community? This is after some work done by the U.S. Fish and Wildlife Service. So we have two pieces of information here. One is that we have a species accumulation curve. Two, we also have an estimate of the total number of species in the system. And so we can fit a line to this species accumulation curve and extrapolate it out to the total number of species in the system, or estimate to be in the system. And then look to see how many, or estimate how many samples it would take to sample 95% of the community and 50% of the community. So we did this for both the traditional gear approaches and the light gray and the combined eDNA approach that we use. And we did this based on a chow estimate of total number of species or jackknife estimate of total number of species. And what I'm showing you here is on the Y is the number of collections necessary to sample 50% of the community and 95% of the community. And in both cases, eDNA sampling approaches can sample 50% and 95% of the community in fewer samples. So, given what I've shown so far, it's to me not surprising that eDNA approaches effectively detected aquatic invasive species in these systems, and that we've got 3 different aquatic invasive species in the vertical panels, and the horizontal panels are the different lakes, and the y-axis is proportion of samples, and that in many cases the aquatic invasive species are observed in a large proportion of the samples, and that there are some cases when the aquatic invasive species is only observed using an eDNA approach. So, in summary, we found that more species are observed in a single eDNA sample on average compared to this multi-traditional gear approach, and that this eDNA approach is effective at detecting aquatic invasive species. And how we see this is that it's not a replacement for traditional gear by any means. It's something that can complement a traditional gear approach, and that they should be used in a comprehensive effort to sample for rare species, whether that's invasive species or threatened and endangered species from the context of fish, or we've actually started to expand this to non-fish taxa as well. So just, I'm new to the area, so I just want to introduce myself to this group. So again, I appreciate you sticking around for my talk. I am interested in studying ecological or evolutionary questions in the Great Lakes using genetic or genomic techniques, and I'd be happy to talk to you about any potential collaborations. So with that, I'd just like to thank our funders, the Great Lakes Restoration Initiative and the Michigan Department of Natural Resources, and there's plenty of field crews that helped out collecting the fish for this study, so we'd really like to thank them. And at that, I guess I'll take some questions.